Hello everyone. Here is the next part of the analysis of RNA-seq data to compare gene expression in breast cancer triple negative cells MDA231 with gene expression in estrogen receptor positive cells MCF7. In the previous parts, I have uploaded FASTQ files from Gene Expression Omnibus GSE 188914. 3 FASTQ files from MCF7 and 3 from MDA231 were submitted to a pre-existing workflow within the geneXplain platform to calculate the differentially expressed genes. The resulting upregulated genes were mapped to gene ontology terms, to diseases and to pathways. As the next step, I selected a functional group of genes curated as causal biomarkers for breast neoplasms, and today I will apply the upstream analysis to identify the master regulatory molecules. On the start page, in the category Upstream Analysis, I click and open the workflow Upstream Analysis with Feedback Loop. I need to mention here that to be able to apply this workflow, I have got a license for TransFARC and TransPath. As we can see here in the workflow, in the first step, the input gene set is subjected to the method site search on gene set, which predicts transcription factor binding sites in the promoter, applying the TransFARC matrices. In the second part of the workflow, the regulator search is performed on the identified transcription factors with the help of the TransPath hub. And in this way, the network of the TransPath database is analyzed. Master regulators that are upregulated are further filtered and annotated and saved in the output folder. Just for us to remember, I am searching for master regulators in breast cancer triple negative cells MDA231 as compared to the estrogen receptor positive cells MCF7. To execute the workflow, I input the genes upregulated in MDA231 and known causal biomarkers for breast neoplasms as the yes set. I select log-fold change the column to calculate expression values. For no set, I select the non-changed genes. All other parameters are kept as default, and I press the button Run Workflow. The workflow starts executing. After the workflow is completed, let's have a close look to the results. There is a folder which is created, which includes several tables and diagrams. The site search result is the result of promoter analysis, and it includes the site models which are overrepresented in the yes set versus no set. Within the workflow, these models are converted into a list of transcription factors. Let's have a look which transcription factors are significantly overrepresented. The analysis done suggests that these transcription factors are responsible for gene regulation in breast cancer triple negative cells. Important point is that these transcription factors with overrepresented matches were used as input in the next step, the master regulator search in the network. The next resulting table is the master regulators with expression values. Each master regulatory molecule is characterized with a score, Z-score, FTR, and rank sum. In this table, we can see the master regulatory molecules that are upregulated in triple negative cells versus ER positive cells. If we sort this table by log-fold change, we can see the top upregulated molecules. CD40 is upregulated close to 10 times, GR is upregulated 4.5 times, E2F7 is upregulated 3.6 times, and E2F1 is upregulated to 2.9 times. If we sort this table by rank sum, we can see E2F1, GR, and E2F7 molecules on the top. Here I open the diagram E2F1 in the workspace. I can see the molecules ELK3, cyclins, DP1, molecules in deep red color as highly upregulated, and molecules like PAC6, FLI1 as downregulated. As we can see on this diagram, E2F1 is responsible for coordinated regulation of the network below. As the gene encoding E2F1 is upregulated itself by 2.9-fold, this is the positive regulatory feedback loop. There are at least several publications confirming the role of E2F1 in triple negative breast cancer cells. I mention two of them here. For example, the paper published in Molecular Oncology confirms the role of E2F1 in regulation of AMP32E, which in turn promotes cell proliferation in triple-negative breast cancer. Another example. In this paper published very recently in Cancer Biomarkers, it demonstrates the key role of E2F1 in regulation of cyclin A and other genes that define the cell cycle progression and thus promote the growth of triple-negative breast tumor. To summarize, in this part I showed upstream analysis of the genes expressed in triple-negative breast cancer cells. E2F1, GR and E2F7 are some of the master regulatory molecules which potentially play a role in triple-negative breast cancer cells. Thank you for watching and do subscribe to get more updates.